information multi-domain data, and you'll have to explain to me what the difference between multimodal and multi-domain data is. After the seminars, we can all go back to the coffee machine and the instant coffee. So, Susan, please.
Okay. I guess I'll start by saying multimodal for me, coming from a statistics background, I see a density with several bumps, and so this doesn't have anything to do with that. So multimodal, we don't use that word, we use multi-domain. And some of the Dutch use multi-way data, different cultures use different, but I definitely don't use multimodal because of the density background, but it's different types of data. So I'm going to, for me, networks never come on their own. So the data that I work on is always very complex. And when I talk about complexity, it's heterogeneity at many different levels, which I'm interested in. And so this is a picture where we have sort of a graph and clusters and different textures, and this is the type of heterogeneity I'm thinking about. Now, in the particular, I'm going to teach you something, at least not about the mathematics, but I'm sure that I know more about the microbiome than most people here, so I'll teach you a little bit about what we do when we study the microbiome. And in particular, I'm interested in the effect of antibiotics and diet perturbations, but also in studies that try to predict preterm birth by taking bacterial samples. So the challenges that we have, and I'll start off with the heterogeneity of the data, but it's also what we call the data integration. So we have sources of information that come in the form of graphs and networks. Sometimes we're trying to estimate graphs and networks, but we always have other structures. So for instance, in the pregnancy data, we have time series and we have longitudinal data. And many of the manipulations that we make it so that we are almost in a multiple testing situation, but we can't correct for it because it's not, I would say, formalized in a way where you can actually say, I did this number of things. There's a combinatorial explosion of the number of possibilities. So we counter this with having a reproducible workflow. So let's start by the heterogeneity. And the first thing is that for me, sometimes we have a response variable. So trying to predict whether what the length of the pregnancy is a typical case where you do have a response variable that you're trying to understand. But sometimes you don't. So it's cases of unsupervised. You're given a whole set of networks and you're given covariates and you're just asked what's going on. And you're not told that you're trying to predict anything in particular. So that status of the variables is sort of an important aspect of what we do. And I like, all statisticians know how to deal with regression and supervised situations much better than we do with, what's going on type of situation. On the other hand, for most of what we have hidden or latent variables, things that are measured explicitly. And then we have these underlying groupings or categorical variables, clusters, which are not observed, or we might have gradients or low dimensional structures, which are not directly measured, but are present in the data. We have different types of data. We have continuous data and binary and categorical data, which statisticians know how to handle very well. But then on the other hand, we have graphs and trees, which we don't handle so well, and images and rankings, spatial information. So we have many different types of data. And then most of biological data is not IID. So we can't pretend that we're in the, we have IID data, so either we have spatial dependencies or longitudinal dependencies, but we have a lot of dependency. And we're combining data from different technologies, and that also brings in various kinds of batch effects and difficulties. So let me talk a little bit about the data itself, the way it comes in. The microbiome is a collection of bacteria, communities of bacteria. And we measure one particular gene in each of the bacteria in order to have a fingerprint for which species are present. But we also have transcriptomics, so we also have things which are measured in terms of gene expression, so many, many different genes, regardless of which species they belong to, they are interacting. So that's why we have systems of communities of bacteria which are able to metabolize certain chemicals, and one species on their own can't do it. So the transcriptomics are important in the structure. So mostly people think bacteria are harmful. 99% of them are not, and we're all healthy because of bacteria, so it's definitely something that we live with in symbiosis. So the genetic signature comes in the form of DNA, so we have, and this is where this multi-domain data part comes in, we have contingency tables of counts of reads of DNA of different species. We have counts of gene expressed, measured by mRNA, and we have proteomic data, which is very difficult because it very often comes in unlabeled form. So we see mass spec or proteins which are present, but we don't exactly know which ones they are. And we have a lot of clinical multivariate information about the patients, their clinical history, their weight, their medication that they're on. And some samples come in with environmental location. We have information about nutrition and time. And then we have a lot of domain knowledge, which is very often in the form of graphs or networks. And metabolic networks are well documented. Chemical ones are known. Phylogenetic trees are all, in my context here, they're all well known. They're documented, so we know what the family relationships between all these bacteria are, up to a little bit. And then we have information about gene ontologies, which are also in the form of trees. So for me, some people like to slice up the data and say, this is not real data, this is metadata. I just say everything is data, and there is no such thing as metadata. That's something that the NSA is trying to make you believe. So the reason I got into this is definitely because of the phylogenies. So I started studying phylogenetics more than 20 years ago. And it's true that for every location in the body, we can plot the presence of a bacteria on the original phylogenetic tree between all the bacteria. So the skin, for instance, there are clumps which occur in the tree. And I'm going to try without turning this off. You see here, we have this clump of species which are present, which are in a specific area of the tree. On the other hand, for instance, the gut microbiome, we have all these measurements of presence which occur right across the tree. And then here it's quite sparse across the airways and very diffuse. So there are many different ways of taking the tree information into account, but it's definitely something that we can't ignore.
Excuse me, is that, are these supposed to be the same tree, or are they different trees?
Yes.
For different locations?
They're all aspects of the same, it's made from the same reference tree.
Yes.
Sometimes they've ignored some of the trees, so they don't look the same exactly, but there's a gold standard tree you can imagine with 10 to 20,000 bacteria, and that's known. And so they're different parts of the tree. But
and the red there are the ones that are in the skin.
Yeah.
Is this, yes.
So the bacteria that lie in different places in the body lie often in different parts of the tree. It's not completely true. There are bacteria which you
Is
find.
this from a single person or some kind of average? Because I guess it may be individual variations.
Oh yeah, there's an enormous amount of variation. So this is just one sample. You just take one person,
sample,
one
yes.
point, one sample, and then this is what you saw. Yes. And then it changes all the time. Also, we have terrible technology, so it's terribly noisy. But and there's contamination all over the place. So. So the questions that come up, or really, we have a consortium of people who work on this, and it's interesting because, in some sense, this is a place where ecology meets medicine. So there are methods that came from ecology which are quite qualitative, which were ordination and measures of diversity and environmental influence. And then people coming in from biostatistics and clinical statistics and clinical studies, they just want to do a test and have a p-value, and they want to do a clinical trial whether, you know, this type of treatment is having an effect on this species or not. So it's very varied in perspective. I work with people who come from systematic biology and statistics, and we also do a lot of visualization. And of course, the network science comes in because we do, you'll see, we're trying to understand the microbial communities as they evolve under certain pressures. So that comes in quite often. So just to say how I see the data, I'm very anchored in using reproducible research tools. So I use R, and it has an enormous advantage. It's an object-oriented framework. And so you can do object-oriented statistics where these objects are composed of many different pieces. So you have the contingency table of the reads of the different species on the samples, and then you have a relationship between the different species through this phylogenetic tree, which is also sometimes written down in terms of kingdom, phylum, and classes. That's what we call the taxonomy table. And then on the other hand, we have all the variables that are measured on the samples. So you have these medical variables, BMI, and so on, which are measured on the sample. So you have these different sources, and you can put them into these heterogeneous objects through what are called S4 classes in R, which are very useful. From a mathematical viewpoint, I think that even if storage of networks and graphs as matrices is very cumbersome, we think of them mathematically as matrices. So the main thing is that we don't just have one matrix. We have a whole set of matrices, not only because we have longitudinal, but we'll have different metrics. We'll want to take into account distances between rows and columns using different metrics. So we have many different matrices. And I'll talk a little bit about what I like as an approach to that, which are called duality diagrams, which capture very well that we have different types of perspectives. One is to look at samples, and the other is to look at the taxa. And so one of the tools that we've developed, which is in some sense a tool for, I'll show it to you online just to see, so that you can see a typical case. So this object-oriented framework in R is not what directly the biologists use. We made a shiny front end to it where they can take networks. And so here, this is a network between different samples, and they can interact with their networks. This is a network, in fact, which was a co-occurrence network, which is created using a Jacquard similarity. And we have nodes, which are the sample nodes, and we'll see later on that these nodes are actually meaningful, and we have overconnection in some sense of nodes of the same color. And you can see this in here. And then actually this one sort of disappeared, but it's out there. I don't know how to make it come back, but this is very useful for interacting with what we call hairballs. But so as I was saying, there's this duality. So on the one hand, you have the co-occurrence network, which joins two samples if they share a lot of species in common. And on the other hand, I could look at taxa. Uh-oh, all right, my connection died, so now I've killed everything, but it's all right. All right, I'll give up on that, but technically I should have been able to do exactly the same thing, but instead of taking samples, I would take the taxa and look at taxa that were present together, co-occurring in a certain number of samples, then that would make it then be connected. And that's how we see the communities. So there's this idea that, in fact, there are two perspectives. We have these dual perspectives. One is on the rows and one is on the columns, but we have networks on both. And so here are some of the networks that come up for me in the applications, and I'll show you some of the ways we work. So there's the co-occurrence network, that we have samples that are connected because they have a lot of the similar species. We have geometric graphs that are defined by a threshold on a specific distance. And we also have the notion that many of the ecology methods are based on similarities. And these similarities are often, can be considered to be weighted graphs. We manipulate them as matrices, but then afterwards the representations are often as networks or graphs. And the phylogenetic trees themselves give us a metric between the different species, because you can consider the patristic distance. And we'll see that we can also integrate graph distances into the way that we look at the data. So we can add the information, as I said, between the samples, or looking at the distance between the taxa. And that's what I call the duality principle. And so what this says is, on the one hand, you can consider that you have a distance between observations or samples or statistical units, and that would be the rows of your standard data matrix. And so here I put x1 dot, x2 dot, x1 dot, xi dot, sorry. And then here we have the variables. So these points actually lie in an RP dimensional space, and so you have n points. On the other hand, the variables themselves, they actually lie in the space of observations. And we have both ways of seeing it, and it's very true for the microbiome data, because it's not always the case what's so well defined as they're samples from something. The rows aren't necessarily samples. So we just have this idea that we have this duality. And you could look at things from two aspects. And so it's funny, because in the graph world, it seems that some people say, well, I do graphical models, so I only study the graph between the variables. And then there's another group of people who say, well, I don't talk to you because you do, each of your people, and I'm doing those graphs, and these are so different. But actually, and as I'll show you mathematically, if you have an underlying low-dimensional representation, they're dual and they're actually the same. You can go back and forth. So, that doesn't work as a way of opposing. I like to see them as the same sort of thing. So the other thing that happens is we can actually add metrics or uncertainties to both the observations and to the variables. The standard one on variables is to consider that you do PCA, and then you take, you divide by the standard deviations, and that's equivalent to taking this diagonal metric. And if in fact you have a graphical model with some kind of structure between the variables, you could have a non-diagonal matrix here. And you can take it out, or you can put it in, and that helps in some sense keep all the information together. And here, this is a case where I have weights on the diagonal weights on the individual observations, and that can be due to the observations actually come, as in my data, from sample which are sequenced at different depths. So some of the samples are very well understood because we have very deep, a large number of DNA reads, so they're very precise. And some samples you don't have a lot of DNA reads, so you have a different level of uncertainty on the points. So this gives you the two metrics, one for the observations, one for the variables. And this is now being generalized to kernel methods as well, where you can do this. And so just to come back to something you know well, this is the case of PCA, but we put in these metrics so that they make orthogonality slightly different because you take these weights into account, but it's the same thing. And we have the singular value decomposition and the eigenvalues just for the weighted generalized singular value decomposition. So that's somehow, important. And I'd insist again on the duality, which was something, so Escoffier was my advisor, so maybe I'm indoctrinated. This is the way I learned statistics. That is, this is called the duality diagram. And in this, you can define actually two operators here, V, x prime dx, which would standard variance covariance matrix, and this is nowadays called the Gram matrix in kernel world. But in some sense, this is the duality that you have. And the important thing about that duality principle is VQ, if you go around the diagram in one direction or in the other, they both have the same eigenvalues. So whatever rank is underlying, and so whether you're looking at the observations or looking at the variables, if it's low rank, it comes up in exactly the same way. So that's why I say they're sort of dual aspects of the same thing. We can use D to code up a prior distribution, and of course, the non-matrix form of these operators, this has been generalized, this duality using kernels. I won't go into it. One of the consequences of this point of view is actually quite nice. You can take the characterizing operator of your study and look at how close it is from another one. So if you have multi-domain data and you do a PCA with one and a PCA with the other, and suppose that you have the same observation set, but you have different classes of variables, you can look at a correlation, which is actually an angle. It's a generalization of a correlation between vectors, but it's a correlation between matrices. And so this is it's just a trace in a product that we use, and we call it the vector covariance. And that's just this angle. And here, this is the equivalent in some sense of the square of a correlation coefficient, but these are multivariate. And so you can have two operators. And if you just took one vector for each, then you're back to taking the square of the correlation coefficient. But this is very useful because it allows you to do multi-domain things. And you can show that PCA, for instance, is the study that has the minimal rank, but that maximizes this correlation coefficient between the low rank one and your original study. So this is just the one with the minimum angle, but it's something that generalizes nicely. So I'm not going to belabor too much about duality, except that in the interpretation and going back and forth when we work on the microbiome, it's extremely important to be able to do what we call the biplots. And so in the program that we set up for studying microbiome data, we give a priority to methods which allow us to look at the samples and the species at the same time. We don't want to, we want to be able to interpret everything that we see about the samples in terms of the species, for instance. So we need both faces. And so here we have the taxa, and this is the different sample types. So here we have the samples are here, and they come from different environmental areas. And then here we have the species. So you can see these samples cluster together, and these samples cluster together, and these, but you can see why. You can see which of the species are responsible for the co-occurrences. So I think that keeping both sides of that duality is really important. So as I said, we have these many matrices, and the type of study that I do a lot of, I often put it in this format. So we have, you might have contaminated, uncontaminated, perturbed, unperturbed. So you have a two sample situation, or two treatments in some sense. And then we have this multi-domain data where we have, here this is functional data, so we have a network which is metabolic network. Here we have genetic interaction network. And here we have the taxonomies, which is the tree. So we want to combine these, and often they're time series. So you can see that the level, the networks come in, but there's a lot of other things going on that we have to take into account if we want to use all the data. So our main workhorse, in some sense, apart from using different fancy metrics and then doing PCA, is also to use multidimensional scaling. So Schoenberg's method, 1935, the idea was you had a matrix which are of positive numbers, symmetric, and you can ask, what would make those numbers Euclidean distances, what coordinates, and under what condition that happens. So he did it the exact case, that is, you have to consider that if you had your original Euclidean matrix with coordinates and you applied a centering to it, and then took the square, the product, the matrix of the squares of all the distances here, and if this matrix is semi-definite positive, then you can say that the original matrix the matrix D is a distance between Euclidean points. And what happened was then Eckhart and Young, 1936, proposed an approximate version. That is, you can go actually from any matrix and give Euclidean points which are approximately distant with those distances in a lower dimensional Euclidean space. So this is something that we use all the time, and it's used for all kinds of different distances which integrate the graph structure, and I'm going to show you one of them. This is called, so in the ecology world, multidimensional scaling is called principal coordinate analysis. And so that's why historically this method, which allows you to take into account a distance on a graph and abundances at the same time. So my graph in this particular case, the one I care about the most, is phylogenetic tree. And the distances between the taxa are what's called the patristic distance. So it just means the distance on the tree. And you start off by taking that distance, which we call delta, and once you find a Euclidean space, an embedding for those points, you then take those points which represent the species and you assign weights to each of those species for each sample according to how abundant that species is in that sample. So then you take the center of gravity. Each sample is the center of gravity of a whole bunch of species which have different weights. And so every sample occurs in that space, and then you can do the multidimensional scaling to have a low dimensional representation. So that's a method which is useful because it allows you to use at the same time the distance on the phylogeny, the graph, and the abundances. So that's one way of integrating the two. I'm going to show you a little example. This is work with Julia Fukuyama. And in this case, we had about, this was a preliminary study to a big study we're currently doing where we have patients who are given antibiotics. So we have a longitudinal time series and we're following them before, during, and after they take antibiotics. Actually, we take two series. And this is applied to that. So we have the different species here. So this is the PCOA, the first principle, the MDS that's done on the OTUs. Those are the taxa, the different species as they occur on the tree. And these are all those taxa in a lower dimensional space. And then here we have the coordinates of the samples. So one of the things that you can see is, first of all, there are three colors, and the three colors correspond to the three different patients. So everything about the microbiome, the first effect is the patient effect. So we use a lot of random effects modeling because the big difference is person to person. And then the next thing that we try and understand is what makes a person different when they're taking antibiotics to when they're not. So then we grouped the points between antibiotic stress and not. So these points are sort of in resting, and this is in the situation of antibiotic stress. So this patient, this is the difference between the two. D1, we have this patient, and then F is very resilient. There's not a big difference. So this study actually, the goal of the study is to understand who is resilient to taking antibiotics. Some people are extremely sensitive, and we try to understand and see that difference. So this is a case with only 3. We're currently doing more than 100 people. And we see this phenomena that actually the sort of direction between non-stressed and stressed is very similar for many. There's a whole group of patients for whom that's true. But what was important, or what in the understanding of this was saying we have this graph structure on the species that we need to incorporate into the representations. So, yeah.
So in that plot, so E and D,
yeah.
Clearly they're not resilient.
Yeah.
There's a difference, but F doesn't appear resilient in that plot. But you're arguing that it's
No. I'm saying F is resilient.
I'm sorry, F is
in that plot.
in that plot. But,
Yeah.
but what can you say about it?
Well, I can say the following. I can say that it's 54% of the variability. 54% is quite high. So it would be, you'd have to look at 3D, maybe it's coming out here, and that's what we do.
Okay.
But I'm just saying, this isn't a statistical analysis, it's
Right.
3 people, right? But I'm just saying, this is the principle of how we work. And what's interesting with this method compared to other methods that people use, there's another distance on the tree, which is called unifrac, which is a weighted unifrac is the same as Earth mover's distance on the tree. But that method, you can do a PCOA on it, you can do multidimensional scaling, but you don't have any interpretation in terms of what the species are. And so the added value actually in this study was that we have this plot where we can say, well, the direction here is that through stress, there's much higher bacteroides. And that's the thing that they're interested in. So it's in the interpretation, I guess, is important, keeping all the variables together. Okay, so this is another one. This is another study. It has to do with age. The reason I show this is I'm on the soapbox right now because it seems that the world's adopted doing multidimensional scaling and has decided that principal components and multidimensional scaling plots should always be square. And they shouldn't. It turns out that if you have 3 times or 4 times the variability, you can't interpret directions in a square coordinate space and say, oh, look, these are quite similar as these. So if you have differences on one axis and you want to interpret them, you have to keep all the scaling according to the eigenvalues. And so that's something that seems to have been lost in the textbooks, but so not square plots necessarily. So I talked about the multi-table methods and the RV coefficient, and I just want to say that this co-inertia analysis and this RV coefficient allowed us also to look and combine metabolomic data. So this is something I showed you before, but we had the taxa abundances, but we also had the KEGG abundances for the different KEGG categories. We had mass spec data, and we were able to do this multi-domain looking at the links, and we get a correlation coefficient, correlation matrix in exactly the same way as you would if you had vectors. So this multi-domain example, and I'm not going to So I want to talk a little bit about testing because it came up. Several people said to me, well, yeah, we can look at these graphs. How do the graphs and covariance mix when we're doing testing? And I want to show an example of that. So I showed you earlier on when I was doing the interactive thing that you could have different samples and you create an edge between them if they share a lot of bacteria in common. And so this is, we have different types of coloring and identification here. So the color is actually the identification of the mice. And we have gender. In front of the labels here, we have these colors, and then we have 2 litters of mice. And there are various things that come up when you have this type of thing. You want to know, are the mice from the same litters more alike? And this is actually a very troublesome batch effect, which occurs. People didn't realize how bad it was, both for litter and for cages for the mice. So this is using a very simple non-parametric test, graph-based 2-sample test, which came from an idea of Jerry Friedman, who wrote 2 papers on this. So the Friedman-Rafsky test, originally he did it with making the graph he made was the minimum spanning tree in the high dimension. So if you have distances between all the points, it's very cheap to make a minimum spanning tree. Once you have the minimum spanning tree, you can ask, do I have about the same number of hybrid edges, that is edges where you have 2 different categories, 2 different levels of your categorical factor, or do I have clumping in some sense? Jerry's thought of this as a generalization of Wolfowitz runs test, where you do it on the line and you look at the number of runs, so it's the number of edges in some sense. And so this is just a generalization. It's very simple to do this. So in this case, I did a minimum spanning tree on Jacquard dissimilarity, and I only looked at the litter. And if you look at the number of pure edges by simulation, this is what you get. This is the number that you get for the observed here. So we have many, too many pure edges. So it allows you to test, in some sense. You have a graph, and you have a covariate, which is a categorical, and you want to know, is there any structure? So this can also be done, we do it for the co-occurrence networks, and so we have these dual networks, which is either edges between taxa, if more than a certain proportion of samples share that taxa, or edges between samples if they share more than a certain proportion of the taxa in common. So we could do both, and we can look at that. And of course, I prefer, and you already saw this, because Kerry used the Jacquard, and it's very important in this case to be using, when you're doing presence-absence of taxa, especially because there are lots of rare taxa, you should absolutely not use correlation coefficients. So it's funny because a lot of people think, oh, I'm going to make a correlation network, but it's completely wrong for many reasons. First of all, many of the data are given as proportions, and we know that correlations between proportions are biased, and Pearson, studied that very well. But we have to keep away from correlation coefficient. At the same time, we want to count the number of ones that co-occur, and we count the number of ones, and the denominator doesn't take into account the zeros, only the occurrence of places which have occurred in at least one of the 2 samples. So here, this is 0.8, so distance. So then we have this, and this is another example. In this case, it's actually cages. And so you don't see the edges very well. I'm apologetic. This is perhaps a little bit better. So the edges, it's the same graph. So if you look at this, this is the co-occurrence network. So the samples which are connected have more than, say, more than 50% of the taxa in common. And so you could ask yourself, well, do I have any pattern among these? So now I'm going to tell you the secret of the numbering. These are all mice. And the secret is that 7 is the cage, and 2 and 1 are the number, and 5 are the numbers in the cage. Here we have 17 and number 4, this is 16, cage 16, cage 15, cage 17. So this was something I discovered because I was trying to look at something, and I started making the co-occurrence network, and I see this trend, and so I went back to my to the postdoc who'd done the experiment, and I said, is there a meaning to these numbers? And then she owned up to the fact that there was. So the main thing is, it's terrible in this case because there's a terrible cage effect which they didn't control for, and so everything that's going on is because the mice eat each other's poop basically, but not very interesting. That's something that biologists already knew, but somehow they didn't think to control for it. But so in this case, we did the test, and of course it comes out significant. The different versions of the test are implemented in a package that Julia Fukuyama wrote. It's available on GitHub. And we have, minimum spanning tree test, k-nearest neighbor graph, geometric graph with a threshold. All of those are implemented. There's a very nice theory now, which has been developed over the last 2 or 3 years by a student who worked at Stanford, Paswa Padacharya, and he has 2 very nice, if you're interested in these tests, these are beautiful papers, first of all about distribution, theory central limit theorem, and then the power of these tests in different situations. So these are 2 papers which are going to appear. They're currently up on the archive, but they're very, very nice math stats. So
in that analysis, is it, I mean, you mentioned nearest neighbor, or minimum spanning tree, or other choices. I mean, are the results very dependent on the graph construction?
No, I mean, mine is gigantic effect, so it doesn't matter what you use. Yeah. The natural one here was the co-occurrence one. When you have very high dimensional data and it's not counts, the minimum spanning tree is the standard one that we use. And that was the standard test before.
Right. Okay, before.
But some people like the k-nearest neighbor graph. But it's just a very useful way of saying, okay, I've got this graph. Now, does it have anything to do with the covariate? Yeah. And it's sort of very nice development. And sort of this theory brought it back. I had a PhD student, Nelson Ray, who worked on this, looking at various data, and he actually did a little science method development on some of the theory, but Pasquale's done a wonderful job of really developing this. Now I'm going to do another type of testing, which is something that we're working on for the microbiome, but I'm going to talk to you about results which have already been published, because I work with biologists. Sometimes I can't talk about the real problem, but it's the same situation. So in this situation, we know for the metabolic networks and the gene interaction networks, we actually know what we call the skeleton graph. It's given to us. So there are databases like String and things like that, which give us a gene interaction network. And then what we have is we can map onto this. If you have a 2-sample test, you can map onto it your p-value or your t-statistic. And you can ask yourself, in this graph, where's the hot spot? That is, where is the place where there's a big differential? Where is the place where the p-values are the smallest or the t-statistics the largest? So this is, for instance, a skeleton graph of the chemokine network. And this was published more than 10 years ago. And we were looking for a subgraph which was changed in cancer patients compared to healthy controls. And we have a scoring function. And so in this case, it's a t-statistic. And you assign that t-statistic. And the problem is actually a NP-complete problem where you have to try and find the subgraph with the highest. It's like a knapsack problem. A subgraph with the highest score, and we do it with simulated annealing, and then we look at the results using permutation and bootstrap type test. But that's a sort of useful, I thought I'd say this because it's a useful, people were saying, well, how do you test things on graph? You can often map results if you have the existing skeleton graph. Of course, the idea here is that we are gaining strength by looking at a neighborhood, but if you have missing links in your graph, you're not going to see it. So I always advise people, when you're making your skeleton graph, be more loose, don't make the complete graph, but be loose about what your criteria are for taking the graph. The last part, I'm going to talk about an application which I said I was working on pregnancy. So we're looking at microbial communities over time. And this is a case control study of about 49 pregnant women. So we had originally, the original sample was 40 women, and they're measured all through their pregnancy every week, as soon as they're declared pregnant, and then on until they give birth. And what we'll look at, although we looked at the oral, the fecal microbiome, we're going to look at just the vaginal microbiome and see what happens. And what we had to do, and I'm sliding a lot of things under the rug because there's a lot of heteroscedasticity that we had to deal with, and I'm not going to talk about that. I'm just going to talk to you a little bit about the sort of the network aspect the graphs involves, and especially the communities. This paper was published in PNAS last year, and there's an extensive supplement in R with all the data and all the analyses so that you could go through every step. So the questions that we were asked by the biologists were, they wanted to know where there were community state types that were the same as in previous studies, and whether they were stable across pregnancy. And of course, they wanted to be able to know whether there were certain types which predicted preterm birth, because that would serve as a biomarker for preterm birth. So, so this is a plot that I made this morning because I thought that you would relate to it better than some of the other plots I made. But what we have is all the samples. So this is the dissimilarity between samples here is computed using what's called Bray-Curtis distance. And so these are very, very similar. So we have, here's a community in some sense, a group of samples which are very similar. This is a second one. And so up there I've put colors which don't come up as well as I was hoping, but they're kind of washed out here.
But the row columns are subject or sample?
Subject. So here I put the subject and this co-occurrence they're sharing, you know, and so they're samples. They're actually not subjects. It's the same subject measured over a long period of time, and you'll see that these are all, so there are about 800 here. Okay, and then we have here just a little bar as you go along, and this is whether they gave birth early or not. So these ones gave birth early, and these ones gave birth early, and all the green ones, they didn't. And then what we have also is these different groups. So these are the community state types that correspond 1, 2, 3, 5, and then this is a problematic no man's land, which we're going to declare 4 for the time being, but we're actually splits into 2 subgroups, and which we're most interested in. Why are we most interested in? This is where everything occurs. That is all the preterm birth. So if we look in terms of actual abundances of the taxa, the bacteria, it's actually a much simpler picture because they're dominated in some of the groups. So the Lactobacillus crispatus dominates completely the first group. And the second group is Jensenii. And then here we have Iners, which dominates the third one. And the fifth is interesting because it's a mixture of those two. But here in 4, the main thing is it's not really dominated by anything in particular. So it seems that this group is very, you know, it's like the principle of, you know, unhappy families are all, no, it's the opposite, right? Anyway, they're all different. So this is how the subjects work out. So if I put them back into, you know, every one of these samples where it's measured, and I give it the color which corresponds to its block type, we see this longitudinal analysis. And so we see that actually it's extremely stable. That is, if somebody is green, they're going to stay green for a long time. And this is the length of the pregnancy in weeks. So we have here, around here is when things, this is the limit of preterm is here. And so we have a lot of red and pink. These are the two groups. So we split actually that group 4 into 2 pieces, but still that accounts for a lot of the preterm. So there's definitely an association between the type and the preterm birth. And this was actually validated. So we had 40 subjects. We did this analysis, and then we looked at particular taxa within this group 4, which could be extra biomarkers. So being in the group 4 is already bad news, but then there are certain biomarkers within that group. Gardnerella is one of them, and that's definitely something which is confirmed now. So we were right. And so we modeled this with a little Markov chain where you can see that the stability, of group 1, for instance, very, very high. That is, most of the time, if you're in group 1, you're going to be. So this countered what was thought. That is, all previous studies had thought that there was an evolution through pregnancy, that there was a instability, that is, you change groups as you advanced your term. And so this counters that. That is, most of the, and that's very important because it means that if you're in this group, at any point in the early pregnancy, you should be looked after. And it doesn't matter, that it's 2 weeks before term or a month before term. You can even detect this early in pregnancy, and it's still a sign, a good early diagnostic test of problems. So that's quite, that was a good. So the preterm prediction and the stability, I can show you a little bit. Don't have much time, but you can go and look and so more of the details of actually everything that we did. So this is what I said about reproducibility. I can make any p-value be significant because I have a lot of training, and I know how to transform data, and you leave out 9 outliers, and you do this thing, and you're home. You've got yourself a significant result. So I can't trust p-values. I don't believe in them. And the only way I think that we can do honest research and that we can build on each other's work is to publish everything that I did. So in all my papers, the supplementary material allows you to reproduce everything. And so here you have code for every plot and figure that appears, and so it's by exhaustion, right? If you could follow everything that we did, then so these are all the other body, we did saliva, the tooth and the gum, and then you can see all what happens before and after, and then there's. So you can see that, for instance, if you look at the number of samples, whether or not they're preterm. These are the preterm ones. There's many, many more from group 4, and you can do various tests and find out which were the taxa which were responsible for that. And we did all kinds of other things which I'm not going to go into, but I just wanted to say, if you're interested, you could actually go back and do some of the analysis yourself. What we saw, and we did this prediction first, and then we took 9 subjects with slightly about 15 time points each, and we did a validation on those 246 specimens from these 5 women, and we were able to confirm that the Gardnerella and the ureaplasma abundances were elevated for them as well, significantly. And so our prediction after the fishing expedition seemed pretty good. And new data are now coming in where we're doing many more women, and this is still true. So, and it's not causal. So I saw it was written up in the press, and God knows what they're telling these poor women to use special kinds of soap. It's not causal.
It's not causal.
No.
That was worth mentioning.
No.
Yeah,
I mean, we haven't proved causality, right?
Oh, but right, but now you're saying it's not causal?
Well, we don't know.
Oh,
I mean,
okay.
but I prefer to say we have absolutely nothing about causality, and it's for me it's a marker of something else.
Right.
Levels of inflammation, hygiene, all kinds of things which are going on, but
Right, but
very far from me to say that these bacteria are.
right, but you're not claiming that it's not causal.
Well, yeah.
Okay.
All right. I have my opinion on that. But.
And it is that it's not causal, which is that's a pretty big deal. Because looking at
No,
the plot,
my co-authors won't be happy if I say that because they're trying to sell it that maybe.
okay.
Okay.
You're on
I
microphone.
love it.
Some of them. Okay, so just to wrap up some of the things I talked about, and if you're interested, we have various programs, and you can ask me more about this. I'm here all next week, and I'm going to be in the UK for another 2.5 weeks. So the current, we have this interactive graph visualization, which uses D3 and allows you to pull apart networks with all kinds of covariates. I didn't show you how all the filtering and various bells and whistles work with that, but for us, this has been a sort of a gateway drug to get some of our biologists using R and thinking, oh, this is really cool, now I can learn it. But so we also have many ways of testing how you map factors onto graphs and hotspot detection. We're doing threshold and sensitivity testing because we have all this code and we make very different threshold and see how robust things are. And we have the code available. So if you want to look at some of the code in particular for the Friedman-Rafsky was published in this F1000 research, which has always code that goes with it. And the work in progress we've done some uncertainty propagation across ordinations and clustering, some of which I've done with Sergio and Boyu, who are here, but we're also integrating mass spec and NMR data and doing the diagnosis. So this is a picture from a paper from Boyu. We changed the names. Instead of being 1, 2, 3, 4, 5, we have 1, 2, 3, 4A, 4B, but this was also a plot in a paper where we were trying to do this uncertainty propagation. So we have a model, which is a Bayesian non-parametric model, where you can see that instead of just having ordination points, you have confidence regions. And it tells you that these points, for instance, are very uncertain. You have a much broader, and that's because, in fact, the sample length for them are not very high. So that's very useful when you're trying to evaluate whether you really have clusters or not. And so that's all. So thanks to all the statisticians who wrote code that I used. And thank you very much to my collaborators. That's okay. Any question?
I have a quick question.
Yeah.
Just, how long did it take you to develop the kind of relationships with domain experts to be able to do what you wanted to do, or to, I mean, we've heard a lot in applied talks over the past few months of the program about what a great amount of time it can take to invest in working with domain experts in order to be really able to make progress and sort of, aim publications in great journals like Nature and Science and things like that. Just wondered what your experience has been of those types of pieces, aside from the...
It takes a long time for them to trust you. And I don't think it's their fault. One of the things is these journals that we talk about, they hate new fancy statistics. So if you're using a new method, it must be that there's not very much signal in your data. So that's...
Or you wouldn't need it.
Yeah, that's right. So that's the standard. So they don't want to work with you because you're very slow. So for instance, I worked for a very long time on the problem of confidence regions for phylogenetic trees. And the problem came in, somebody asked me, I have a quick question, I'd like to do this. And I thought about it. And I gave a sort of dirty answer, which took weeks. And it took me 17 years to solve the problem. I did solve it, but they don't have time. So you have time. So you have to work on two time scales. And when a problem comes up, it's usually, and you can't solve it, it's probably a pretty interesting problem. But you can't tell them, oh, wait, wait for me to solve it. You have to give them the wrong answer, and so that they can make a nice, their thing that they want to do in 2 weeks, because they don't give you more time than that. But the only way I can work is I have very established collaborators. So with David Relman, we have a big, it's called Transformative Research NIH grant together, and he completely trusts me, and he knows that I'm slow, but every time, I go back, I dig in, and I've done a lot of work to improve his data quality, which wasn't statistical very much at all. But I made all these tools, and his whole lab uses all the tools, and then they give me feedback. So it's a very long term. It's very rare that they come in and you say, oh yeah, I know how to do that. That's just like, this is it. If it was simple, they have pretty good people in their lab. They talk to people. They have students, postdocs. So if it was pretty simple, they probably know how to do it. But the only thing which is really worth saying, because we were talking about it with Kiri earlier on, is that it's very hard to work with people who have bad experimental design. So, the batch effects and the things like that. So getting somebody who is working with you from the outset, and you design the experiment. So for instance, for the longitudinal study of antibiotics, the standard was, and most people do, I take, a sample every week, and then for this, and that's wrong. That's a wrong sample design, because when you're doing a longitudinal study with perturbation, you want to take a lot of points around where it's going to have high variance. And so, but unless they come and see you before they've run it, you get lousy, they're taking every week, and all hell is broken loose during one of those weeks, and you don't have. So I made it so that I made the design of the whole thing so that when they're doing the perturbation, and they say, well, no, all the software wants you to have, and I'm going, no, you want to have a lot
more data can't be better, right?
No, but it's not more. You have to spread it out. I mean, they give you the same number of samples, right? Of course, more data is always better, but you have to explain to them if it's in a plateau situation, maybe you don't need to do every week. But those are the kinds of things which you can only do if you're working with them. And most of the time, they have to write a grant, and they have to do a power analysis. I don't know how it is in the UK, but most of the people I meet, they come to see me, and I don't know, there's a wonderful video on YouTube, which is, I always use sample size 3, right? I always use sample size 3. And, but they need to do a power calculation, so you can already give them feedback at that point when they're writing the grant. So, but this work actually, combining graphs and trees and things like that, she's now associate professor at Berkeley, but it started with Elizabeth Purdom's thesis, and we've done a lot of developing. And she is now working on, she's just studying back on the microbiome and doing sorghum. She did cancer for 10 years, and she's got back to the microbiome. So anyway.
More questions?
No questions. Okay.